Hi, I'm Lynn Fang, a community soil scientist at LA Compost. I help start our community soil health and microscopy education initiatives.
And I'm Maggie, the Northeast LA Regional Coordinator for LA Compost. LA Compost is a nonprofit that works to provide composting access and education across Los Angeles County. When we aren't composting or collecting food scraps at a local farmer's market, you can often find us at the Microscope, educating community members about the benefits of composting and how decomposers influence soil and planet health.
In this video, we will provide a quick how-to on getting started with soil microscopy so that you can learn about the microbes in your soil and the ecological benefits they'll provide to your community.
So, why should we be curious about soil microbial ecology?
Soil microbial ecology teaches us how different types of microbes within an ecosystem build healthy soil. By looking under the microscope, we can see that soil and people are connected through a vast world of microorganisms. As these microbes live, eat, die, and decompose, they transform our food scraps into energy and nutrients, building healthy soils that support all life on our planet. Ultimately, learning about soil microbial ecology can help us make better compost, grow healthier food, and conserve native habitat.
Surprisingly, while the soil microbiome makes up only 0.5% of the soil, its health influences many of the soil ecosystem services that we rely on, such as carbon sequestration from our atmosphere, regional climate regulation, groundwater filtration, growing healthier food, fiber, and fuel, nutrient cycling within the soil, sustaining cultural heritage, and supporting community well-being.
The compost food web contains organisms that live everywhere in the world around us, even in our own bodies. Many of these microbes are crucial for composting in their roles as decomposers. Decomposers can be broken into categories based on their role and function in the ecosystem.
Primary decomposers, like bacteria and fungi, decompose our food scraps and other organic matter. These are some of the most common organisms you will see under the microscope. Bacteria and fungi are eaten by secondary decomposers, like protozoa and nematodes, who then excrete excess nutrients that contribute to nutrient cycling, soil structure, and soil organic matter.
Tertiary decomposers include microarthropods like mites and larger organisms like rotifers and tardigrades. Mites shred organic matter into smaller particles and help to mix and aerate organic material. Rotifers and tardigrades live in high moisture conditions and feed on organic matter, algae, and other small microbes.
Okay, so let's dive deeper into the microorganisms that you can expect to see under the microscope. Bacteria. Among the primary decomposers we mentioned earlier, bacteria are very active in the earlier stages of decomposition. They facilitate the fermentation of food scraps, decomposition of carbohydrates and fatty acids, and suppression of soil pathogens or disease-causing microbes. We typically see three categories of bacteria which come in different shapes, including round, cocci, rod, bacilli, and spiral-shaped spirillum. Some spiral-shaped bacteria, like spirochetes, are pathogenic and can cause illnesses such as Lyme disease in humans.
Actinobacteria. Another type of bacteria is actinobacteria, which are found everywhere in soil and compost. They also dominate the hot stages of composting and are responsible for the sweet, earthy smell of soil that we love. Individual cells of actinobacteria link together to form long filaments, similar to many fungi. This characteristic causes them to initially be categorized as actinomycetes, with the root word myco, meaning mushroom or fungus. In a hot compost pile, actinobacteria can be seen as a white and gray, ashy or powdery coating, and they provide a great visual cue that your compost is reaching thermophilic temperatures.
Fungi. Fungal networks weave through the soil in web-like structures called mycelium. They specialize in breaking down tough, woody materials and plant cell walls, building strong soil structure and aggregation. Under a microscope, fungi appear as brown or white strands that branch, weave, and connect. They can be identified by their smooth, uniform cell walls, which are different from the irregular, uneven edges seen in plant material. They are also distinguished by their septae, or internal cell walls, which look like tick marks and demarcate individual cells. You may also find fungal spores, which can appear as small, dark ovals, almond shapes, or even boomerangs. Fungi grow slowly, and their networks are easily disturbed, so their presence indicates a more mature and undisturbed ecosystem.
Protozoa. Protozoa are secondary decomposers that feed mostly on bacteria, releasing nutrients and plant growth-promoting hormones back into the soil. They also thrive in and indicate high moisture conditions. Testate amoeba have a shell with a distinct almond or balloon-like shape and an opening on one end. Their shells sometimes have a subtle pattern of tiny scales. Flagellates have one or two long whip-like appendages that they use to propel themselves around. Ciliates have tiny hairs around them, which allow them to move quickly in moist environments. Finding many in a sample can indicate anaerobic conditions.
Nematodes. Nematodes are classified by their different feeding groups: bacterial, fungal, plant root, predatory, and omnivorous. Because they consume various microorganisms, they play a large role in healthy nutrient cycling. Their presence also indicates a high level of moisture and microbial diversity within a sample.
Microarthropods and mites. You may also see some invertebrates such as mites under the microscope. They feed on smaller microbes like fungi and nematodes. Their movement within the soil can create more airflow and can redistribute organic matter, while their excrement provides additional nitrogen and nutrients.
Tardigrades. In forest samples, especially mosses, you may find tardigrades, also known as water bears or moss piglets. These eight-legged creatures are whimsical but mighty and known for their ability to withstand the vacuum conditions of space. They are indicative of aquatic environments and can be rare in soil and compost samples. Occasionally, we have seen tardigrades in compost samples, particularly ones that are higher in moisture. Tardigrades feed on algae, plant material, nematodes, and other smaller microbes.
Minerals and other materials. Minerals resemble shards of crystals or salt particles and come in lots of different shapes and colors. They are also very important soil indicators, as they can make up to 40 to 49% of soil composition. Microbes can colonize mineral surfaces and utilize them for nutrients, breaking them down into smaller particles.
Organic matter. You may also see chunky clusters that are darker brown in color. These are organic matter complexes where microbial secretions glue organic and mineral components together and offer a storehouse of nutrients, carbon, water, and microbial habitat.
In addition to microbes and minerals, you may see things like plant material, segments of insects, air bubbles, cracks in your slide, or countless other things. You never know what will come up on the microscope. And if you have questions about your findings, we recommend reaching out to fellow microscopy enthusiasts for a second opinion.
But what even is microscopy? It's okay if you can't say it the first few times. Microscopy involves using a microscope to observe and study objects that can't be seen with the naked eye.
Studying a soil sample through microscopy allows us to visualize the microbial profile present in a sample and consider whether it supports the targeted end use of your soil or compost. It also offers an opportunity to reflect on how your practices influence the overall health of your site. A microscope uses a convex lens where both sides of the lens are curved outwards. When light reflects off an object and passes through the convex lens, it makes the object look much bigger than it is. The microscope we use for exploring these soil organisms is known as a compound microscope and provides magnification up to 400x. The setup we use has objective lenses that magnify at 4x, 10x, and 40x. Because it is a compound microscope, the eyepiece provides an additional 10x magnification. So our total magnification with each of these objectives is 40x, 100x, and 400x. At LA Compost, we use the Soil Food Web Kit microscope set from LW Scientific. This microscope is called the Revelation 3, and the kit comes with a mini-vid Wi-Fi camera attachment.
This camera attachment uses a Bluetooth connection to display our live microscopy on a phone or tablet with the LW Scientific app. This allows us to use the microscope in group settings and even on projectors in the community gardens where we compost. You can connect to the microscope camera by scanning the QR code on top of your camera. Now let's get you started on learning about the parts of your microscope. You can adjust the horizontal distance between the eyepieces. Start by moving them farther apart. Look through them and move them closer together until you see one image.
This part is the stage. The stage clip is where you place and secure your sample slide. You can adjust the slide position with the knobs under the stage, known as the XY axis controls. The top dial moves your stage and sample when viewing vertically along a y-axis, while the bottom dial moves it horizontally along an x-axis. There are two levels of focus on the microscope, a coarse focus and a fine focus. Typically, we start at the lowest objective, 4x. When you place your sample, you will orient the stage so that the light is shining through the cover slip. Use the coarse focus to bring in a rough focus. Then the fine focus is adjusted to further clarify the image.
You can adjust the amount of light coming through, and there is also something called a condenser, which can provide more dark field or shadow illumination. This helps with illuminating outlines of organisms. You can also control the brightness by adjusting the iris diaphragm. Now let's talk about gathering your samples. You can use a hand trowel or shovel to collect about a quarter cup of soil or compost. If you'd like to collect a representative sample, you can also collect small scoops of material from several different spots in your sample area and mix it all together. Be sure to clear away any mulch or leaf litter covering the topsoil. Keep your soil in an airtight container like a mason jar, Tupperware, or Ziploc bag. Don't forget to label your sample with a date and description. It can also be helpful to take additional notes about the environment that you got the sample from. You can hold samples for up to 2 weeks after collection.
When preparing your sample, add 1 milliliter of the soil or compost sample to the test tube. Try to avoid larger particles like sticks and rocks that will obscure your view. Use the plastic pipette to add 4 milliliters of distilled water to the sample, bringing the total volume up to 5 milliliters. Place the cap on the test tube and invert gently 20 to 30 times to mix the solution. Mixing gently helps prevent disruption of the microbial communities in your sample. Let it sit for 10 seconds to let large particles settle.
Prepare your glass slide and cover slip by cleaning them with a lens wipe if needed, and placing them on a clear, easily visible surface. Be very careful when handling your cover slip since it is made from thin glass and can break easily. Use a plastic pipette to collect roughly 1 milliliter from the top of the test tube, just below the organic matter layer. Place 1 drop on the center of the microscope slide. Cover your sample slowly with a cover slip, keeping it from spilling out over the cover slip's edges as much as possible. Use one hand to open the slide holder on the microscope stage, and the other to put your slide into place for viewing. Now you're ready to get started looking at your sample through the microscope.
Make sure your microscope is plugged in and the light is on. You can adjust how the light hits your sample by using the condenser's adjustment knob. Each time you begin a new microscopy session, it is important to start with your stage fully lowered to avoid crashing into and damaging your objective lenses.
For the same reason, always place a sample slide on the stage with the lowest objective, 4x, in place. When you place your sample, you will orient the stage so that the light is shining through the cover slip. Use the coarse focus knob to bring your sample into a rough focus, followed by the fine focus for further refinement. Next, bring the 10x objective lens into place. You can now explore the slide comfortably using the XY axis controls.
There are multiple ways to explore the microscope slide. We can start in one corner and scan the sample in rows to ensure we're exploring every part of the slide, or we can explore more freely to get a general sense of the sample. If you see something interesting that you want to zoom in on, you can move to the 40x objective. It can be more difficult to explore freely at the 40x objective, so it's best used for magnifying something you've already found at 10x.
Sometimes, even if the focus is right, it can still be difficult to analyze a sample if there's too much overcrowding of minerals or organic matter. If your sample is too busy, you can troubleshoot by diluting your sample with an additional 1 to 2 milliliters of water to create more space between the particles. Continue to dilute your samples as needed until it is easier to visualize all of the elements.
Placing the cover slip can also take a bit of practice. If your droplet has too many large particles, it can make the cover slip lay unevenly and you may have trouble focusing. If you pipette too much water onto the slide, the sample will leak out from the sides of the cover slip. This causes a lot of swirling and movement when viewing your sample and makes it difficult to examine organisms closely. If this happens, grab a new slide and try again.
So, besides finding interesting organisms, what else can our microscopy sessions teach us about the larger picture of our soil health? In our samples, we can observe the presence, abundance, and biodiversity of microorganisms, organic matter, and minerals to help us understand the health and quality of our soil and compost to grow healthier plants, nutrient-dense food, and to remediate native habitats.
The different types of microbial communities found under the microscope can tell us how mature and stable or how disrupted our soil ecosystem is. This is explained by the concept of ecological succession, which states that ecosystems naturally evolve over time. For example, an abandoned lot would initially be characterized as a disturbed or pioneer state with low levels of biodiversity. If left undisturbed, this site would begin to evolve towards an intermediate state that allows new plants to thrive. The life cycle and decomposition of those plants would eventually support enough nutrient replacement and soil building to create a mature ecosystem like a prairie or forest.
Let's explore some different soil types and share about how they demonstrate the different phases of ecological succession. Abandoned lot soils. This sample of abandoned lot soil primarily contains bacteria and minerals, with the occasional tiny piece of fungi. This characterizes a disturbed or pioneer ecosystem with low biodiversity and low ratios of fungi to bacteria. It has poor soil structure, low water retention, and low nutrients.
Active compost. Active compost is commonly characterized by high levels of bacteria and protozoa. Sometimes they can have a lot of nematodes, and fungi populations are low to moderate. There is greater biodiversity and microbial biomass, characterizing an intermediate maturity ecosystem. This particular sample features a cilia amidst an abundance of bacteria.
Mature compost. Mature compost is typically less microbially dense than active compost, but has more fungi and more omnivore feeding nematodes. Mature compost samples have greater biodiversity and a higher fungi to bacteria ratio. This sample demonstrates a more stable microbial community with good organic matter complexes. In more high moisture samples, you may even see an aquatic living organism like a rotifer. It's important to keep a report or log of your findings during a microscopy session to guide your future soil stewardship practices and to watch your soil ecosystem evolve over time. All of our soil microscopy participants receive a report of their findings, and when preparing a report for someone, we typically use this format.
We assess the relative abundance of different organisms and provide notes and pictures of each sample analyzed. Although this is a more informal report, it gives participants an understanding of their soil microbiology and next steps they can take to improve their soil health or compost recipes. Microscopy can teach us a lot, but it's important to understand that this type of microscopy will not always tell you the exact species of the organisms you're viewing, and you will not be able to assess the presence of heavy metals. To learn more about these topics, you may need to explore other lab options, such as DNA sequencing or an XRF analyzer.
There's also more quantitative analysis, such as counting organisms or extracting specific groups of organisms from a sample, which require more specific laboratory tools and equipment to do. Our approach is simple, accessible, and qualitative for the practical soil maker to be able to adjust and fine-tune their process. At LA Compost, our community soil health program has blossomed from the occasional one-on-one microscopy assessment with community organizations and local gardeners back in 2020 to a full-blown calendar of monthly events and opportunities for community members to learn, engage, and connect through soil microscopy and lab testing. Learn more about what we're up to at lacompost.org.
It's always a delight to see how transformative and impactful it is for people to see their soil microbes under the microscope. The proof of the invisible life at work in their garden never fails to inspire awe and wonder at the world beneath our feet. Understanding the roles of microbes in our soil ecosystem helps us identify which groups indicate good quality compost and which indicate poor quality compost. Microbes are our guides and shepherds in transforming soil into a living, thriving foundation for healthy ecologies, nutrient-dense food, healthy people, and healthy communities.
Ultimately, there is no one who understands your soils and compost better than you and your community, since you are the ones stewarding it and witnessing its management. Whether you have been an avid sheet mulcher for years or are just starting a garden on a hillside by a construction site, your understanding of the soil's history can be just as important as what a microscope will tell you. By combining any findings from a microscopy session with your unique understanding of your soil, you can develop an effective plan to amend your compost recipe or improve your management practices.
We hope this video supports your journey to healthier soils. From all of us at LA Compost, happy composting!